with high yield topics in biochemistry of the eye. Here I've chosen heparan sulphate as a precursor to the glycosaminoglycans. This is a very important topic. The mucopolysaccharides might cause mucopolysaccharidoses, and this is something that we need to study. So the simple blocks of carbohydrate might give rise to monosaccharides, oligosaccharides, or polysaccharides. Today we are interested with polysaccharides, which are either homoglycans or heteroglycans. Homoglycans are blocks of carbohydrates linked to each other, forming long chains, polysaccharides. They might be either branched or unbranched, and they are just like starch, like glycogen, and some plant polysaccharides like cellulose. These are not really of very big importance for clinical ophthalmology. Heteroglycans are much more important. Heteroglycans are the same as glycosaminoglycans and the same as mucopolysaccharides. And the group of diseases called mucopolysaccharidoses are related to this class of biochemical structures. They are either proteoglycan or glycoprotein. Proteoglycan are mainly carbohydrates, the glycan suffix, but they are covalently bound to some protein component. Protein component is much less than the carbohydrate component. It reaches down to 3% of the molecule, while glycoprotein is mainly a protein which is attached to some carbohydrate. So the protein content of glycoproteins is much more higher than the protein content in proteoglycans. Proteoglycans are either dimethyl sulfate, heparan sulphate, hyaluronic acid, chondroitin sulfate. There are around 30 proteoglycan molecules identified. They include the hyaluronic acid, which has a specific biochemical structure we'll be discussing shortly. And it also contains heparin. Heparin, this molecule that we use for blood thinning, is a proteoglycan. They are more into structural integrity of the eye. So they are found in sclera, cornea, trabecular meshwork, and they are even found in the lens capsule, eye limb, vitreous, and vitreomacular interface. So proteoglycans are formed of what we call disaccharide units, covalently bound to a core protein. Usually these disaccharides are formed of amino sugar, and the other unit is glycuronic acid. So this first part is the amino sugar. It's just either glucose or galactose and attached to an amine group, NH2. This is how an amino sugar is formed. So we form a chain of disaccharides. One of them is amino sugar, and the second one, or the second subunit of the disaccharide, is a glucuronic acid. This is the glucuronic acid structure. So here are some examples: dermatan sulfate, heparin, keratan sulfate. They are formed of these disaccharide subunits. So this is the amino sugar subunit. It's containing amino group, and this other subunit or second subunit contains the glucuronic acid, COOH, on carbon number 5. Besides, all proteoglycans are sulfated molecules except hyaluronic acid. This is a hyaluronic acid and it's not sulfated. Again, it's formed of amino sugar bound to glucuronic acid, but it doesn't contain the SO3, the sulfate group. So hyaluronic acid is the only non-sulfated proteoglycan. This chain, this disaccharide chain, is covalently bound to the core protein. Okay? Parts which are extensively studied for the content of their proteoglycans are the sclera, followed by the cornea. And this is important because this is heterogeneous. There is always heterogeneous concentration of the proteoglycans in different ocular tissues. So, for example, the most abundant proteoglycans in the sclera are dermatan sulfate and chondroitin sulfate. But they are not present in the sclera in the same concentration everywhere. For example, in the peripapillary area, dermatan sulfate is the most abundant. Chondroitin sulfate is more abundant behind the equator, but not peripapillary. This is important because the properties of dermatan sulfate in the peripapillary area allows for distensibility and allows for the biomechanics of optic nerve swelling, papilledema, and inflammation. Heparan sulfate is very abundant in the fetal sclera during fetal development, and this gives higher distensibility features for the sclera. That's why ophthalmos happens. The eye enlarges because of the distensibility of the sclera. Cupping, glaucomatous cupping in infantile glaucoma is reversible. This is important. So after treatment with glaucoma valve or something like that, an 0.7 disc might go down to only 0.3 or 0.4 disc. All this is due to the different content of proteoglycans in the sclera. By advancing in age, heparan sulfate reduces in concentration in the posterior sclera, in the whole sclera, in fact. And in the end, heparan sulfate becomes relatively more concentrated anterior to the equator in relation to posterior to the equator. That's why the sclera loses its distensibility by advancing age. Proteoglycans in the cornea and sclera share many features, like the immunological cross-reactivity with proteoglycans from the articular cartilage. That's why episcleritis and scleritis are autoimmune diseases that are very frequent in autoimmune systemic disorders, because autoantibodies attacking the proteoglycans in the cartilage, in the joints, also attack the proteoglycans in the sclera. There are also other studies on the heterogeneous concentration of proteoglycans in the cornea. So the water content of the anterior stroma is usually more than the posterior stroma due to this heterogeneous concentration of proteoglycans in the different layers of the cornea. And the presence of Heparan sulphate more in the posterior stroma makes it easier for deep anterior lamellar keratoplasty, makes the dissection from this membrane much easier. Diseases involved with proteoglycans are called mucopolysaccharidoses. This is deficiency of the glycosidases, which are the lysosomal hydrolases or the lysosomal enzymes responsible for attacking the proteoglycans. Proteoglycans has a half-life of few weeks, so it's not something that's regenerated every day or every other day. It has a half-life of few weeks. That's why mucopolysaccharidoses are of chronic progressive course. It happens over long period of time, and it's a multi-system disorders. It usually causes organomegaly and neurological symptoms and ocular symptoms. Ocular symptoms in the form of they affect the optic nerve, they do affect the cornea, they cause corneal opacity. They do affect the retina and they cause retinopathies and pigmentary retinopathies. Nearly all of them cause corneal opacities except Hunter syndrome and Sanfilippo syndrome, with all their subtypes. And this is important because if there is no corneal clouding, this is important to differentiate Hunter syndrome from Hurler and Hurler-Schei syndrome. And this is important because Hurler responds very well to bone marrow transplantation, while Hunter does not respond to bone marrow transplantation. It's mainly of neurological symptoms, and bone marrow transplantation does not cross the blood-brain barrier and reverse the changes. Corneal opacities is an important part to mention as well. So a child with central corneal opacity along with hepatosplenomegaly is highly suspicious for mucopolysaccharidosis, especially Hurler syndrome. Bear in mind, I mentioned here central corneal opacity because some children might be born with peripheral corneal opacities and might be having hepatomegaly as well. But these children or these infants with peripheral corneal opacities usually have intrahepatic cholestasis, not mucopolysaccharidosis. Another feature is dysostosis multiplex, which has which shows abnormalities in the development of cartilage and bone, abnormal facies, especially as ophthalmologist, you look for frontal busing with shallow nasal bridge. Is it associated with hypertelorism? No. No hypertelorism in mucopolysaccharidoses. Hypertelorism is usually found in mucolipidoses. We'll be discussing this shortly. Other signs might include abnormalities of hearing, vision, of course we have mentioned all the visual symptoms, cardiovascular system, and mental development. Laboratory tests are like urine analysis for the presence of increased amounts of glycosaminoglycans, and this differentiates mucopolysaccharidosis from mucolipidoses. We can carry out assays of the suspected enzymes deficient in the white blood cells, fibroblasts, or possibly from the serum. A serum sample might be sufficient. Tissue biopsy with analysis of the glycosaminoglycans by electrophoresis. This is a good point for MCQ questions. Electrophoresis and use of specific gene tests or prenatal diagnosis. You can take a sample from the amniotic fluid or chorionic villus sample and do these tests. Now regarding the clinical features, I'll start by the systemic clinical features.